So we're done with this ion exchange chromatography. I hope the students who are there, they have watched it. If not, I will share the this video with you also. Okay. So we continue. Affinity chromatography. This relies on the knowledge of properties of enzyme and proteins. So mainly your antibody and primary antibody plays role here in affinity chromatography. For example, the use of glucose resins to isolate glucose binding proteins, conclavin like A in this case. So glucose binding protein attached to the glucose residues on beads here. And addition of glucose, then glucose binding proteins are released on the additions of glucose at the end. So let's say you have these antibody having these red ions attached to it, which are recognized as antibody, and the blue ones not. So blue ones will remove, and the red ones will be removed later. So some drawbacks that the binding to a ligand antibody can offer be strong and very harsh conditions that are needed to elute the enzyme, and this leads to denaturation. And the non-specific binding may also occur specifically where large ligands such as antibodies are employed.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
Then comes the HPLC, that is your high performance liquid chromatography. In this, the column is very large, tube containing small particles as a stationary phase, and the chromatographic bed is composed by the gel beads alone when they are inside the column. And the sample is introduced into the injector and then carried into the column by the following solvents. So like this, we get the results. You have absorbance at 220 nanometer and the time at the bottom. So you can see 5 different elements has been identified with the help of HPLC. How does this work?
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, the component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
So that was about, I hope my video is still working, about the HPLC. And there is also FPLC, which is a fast performance liquid chromatography. It's a type of liquid chromatography where solvent velocity is controlled by pumps, and pump controls the constant flow rate of solvents and assessed through the tubing from the outside reservoir. Now comes your gel electrophoresis, which is a very famous technique in order to separate your DNA or protein, in which we use three-dimensional molecular sieve. And under the denaturing conditions, the gel can be resolved for proteins of the same size from 10 to over 250 kilodalton, and is one of the powerful techniques. So proteins and nucleic acids, they migrate in an electric field according to the net charge, and for this reason separations can be achieved. In many cases, electrophoresis is coupled to a second separation method to achieve very high resolutions of different proteins. The most popular method is SDS page, where electrophoresis is coupled to sieving device that differentially retards large proteins. So in the gel electrophoresis, this is the simplest in concept but most difficult to achieve, requiring expensive equipment. So buffer of very low ionic strength is passed slowly between two large glass plates separated by a narrow gate. So let's see how it's done. So here you have the glass slides and you put your proteins. So the smaller proteins will go fast and the middle will be in the middle and the large will be on the top. So in this way we can separate. Then you have acrylamide and methyl ethene acrylamide, and then in presence of persulfate they got changed to sulfate, and then you have this chain of polyacrylamide chain. And SDS is coated over the protein so that they could have a negative charge and could separate easily later on. So this is the SDS page stainings looks like. You can also save them for later. Various staining with various timings, and also after staining you can also go for isoelectric focusing gel electrophoresis. On the A you can see high pH, on the left side is high pH, on the right side is the low pH. So all the ions that are present, they will go you have multi-charge ions, and they will find the pH which is the neutral for them, and they will stagnate there. So like for the red they will there, blue, yellow, orange, like this. So this pH gradient is a polyamphylite with many pH values of different 0.01. So you have SDS page, IFE. So protein precipitations, 2D gel electrophoresis. So how we do this 2D gel electrophoresis? So in order to be successful in precipitations of a gel electrophoresis for 2D, so it will explain the technique one by one. So after having the precipitations of our sample, what we do is we go for 2D gel electrophoresis, that we first separate your gels particles on this over this uh, uh, pH plate from pH 4 to 10. Then we separate them over the SDS page. Then we get these small dots here. These are the proteome. All the proteins that are present in particular cell extract, we can check them like this. So let's check how we do this 2D gel electrophoresis further.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the Ipg strip. The second segment will be on how to equilibrate the Ipg strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the Ipg strip. And I'm going to demonstrate this process using Biorad's 2D starter kit. The 2D starter kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, chaps, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 Ipg strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all Ipg strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the Ipg strip into the focusing tray. An Ipg strip has 2 sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip. On the negative side, you'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the Ipg strip is facing down, but you'll be able to read the numbers of the pH range of the Ipg strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before this step. Make sure to label the side of the gel and not the protective backing. To load the Ipg strip into the focusing tray, place the positive end of the Ipg strip into the positive electrode of the focusing tray and slowly drop your Ipg strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the Ipg strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the Ipg strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first, remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the Ipg strip are equilibration of the Ipg strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the Ipg strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the Ipg strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the Ipg strip, turn it over so the gel side is up, and place the Ipg strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the Ipg strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the Ipg strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the Ipg strip. Place the Ipg strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Biorad's Criterion precast gels, which accommodate the 11 centimeter Ipg strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the Ipg strip. Place the Ipg strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the Ipg strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. Do this in one rapid action, side to side. Working quickly, use your forceps to push the Ipg strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the Ipg strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kamasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kamasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kamasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
So that's it for today. The proteomics part is over. I hope my link is also not over. I will share these presentations with you. So that's it for today. So this is the slide. Some more work for you.